Buenos dias, good morning. Today we have a very special guest, Jacqueline Moustakas-Verho, a dear friend of mine. We met in the US in Evo Devo meetings, and she is a maverick, I would say, in the Evo Devo science. She's very well known for her beautiful experiments using new techniques in non-model organisms, her great work on tooth development, on turtles too, the development of their carapace, the shell. Fascinating stuff. Usually as Evo Devo people don't get to publish in magazines like Development. Development requires that your developmental experiments and technique are top-notch. Jackie is one of my friends that has been able to crack that journal and put an Evo Devo research there, which is really top-notch. So I immediately thought she would be a great speaker for our seminars. She did her doctorate in UC Berkeley with the great paleontologist Kevin Padian, and she's now working at the University of Helsinki at Finland. So without further introduction, here is Jacqueline Moustakas.
Thank you so much, Alex. It's really a pleasure to get to present this research to you. So I guess you can see my whole screen. Maybe if I minimize this. And
yes, we see the whole screen.
Okay, great. So I have to admit that I'm grateful that Alex has probably presented his own work to you before, because this Evo Devo field is a little bit complicated. It's quite integrative. So in some ways you can say we're very broadly trained, and in other ways you could say that we are kind of superficial in lots of different fields. But it's the reason why I really enjoy research. So the talk is developmental experiments in organs of skin. Oopsie. There we go. So as I mentioned, I'm studying evolutionary developmental biology. And just as a basic definition, this is how development, which is a proximal causation, impinges on evolution and ultimate causation to affect evolutionary change, and how development itself has evolved. So I'm studying this through morphogenesis. By investigating morphogenesis, so the origin and development of morphological characters or shape. So I'm basically interested in how form comes to be. And shape is something that I think is easy for people to understand and easy to ask questions about, why things have different forms, why they have different patterns, why things also have similar shapes and patterns in different systems. And these are the questions that are driving my research. So as Alex mentioned, one of the things I've been studying are turtles, and specifically the turtle shell. Turtles are easy to recognize because they have distinctive shapes. And why is that? It's because they have the turtle shells. So what is the turtle shell? These are, so the turtle body is basically surrounded by this bony structure. These are plates of dermal bone that form kind of more similar to the bones of the skull, as opposed to, for example, the vertebral column. These have a direct differentiation from mesenchyme to form bone, as opposed to having a cartilage precursor. This is the dorsal part of the shell called the carapace, and then the ventral part here is the plastron. And these bones are overlain by keratinized scales. They're specialized scales that are found on the shell, and they're called scutes. And these white markings here are showing the outline of these scales. They leave impressions on the bone. And then underneath, you can see the shapes of the individual bony elements. So this is a nice system because you can study its development from like an embryology perspective, but you can also consider in deep time through the fossil record.
Jackie, someone is asking whether the pointer, they can't see the pointer.
Oh, you don't see my mouse?
Yeah.
No. Okay. I don't have, I'm not sure if there is a pointer in this system.
I think but I don't know how to use it. Well,
I'm sorry. I assumed that maybe the mouse would actually show, but I guess it doesn't. No.
It usually shows, so that's why I don't know how to use the pointer. Okay, no, never mind. Let's just go on.
Yeah, sorry about that. I'll try to keep it clear what I'm referencing. So turtle shell, the first morphological indication that turtle shell development is occurring is the formation of this carapacial ridge. And it's this lateral protrusion on the trunk of the turtle embryo, shown here. So this is composed of a thickened epithelium overlaying mesenchyme. And the interaction between these two are guiding the development and patterning of this structure. This is a novel structure in turtles that controls the development of the turtle shell. And it provides us with opportunities to study novel roles for tissues and the co-option of genetic networks regulating the structure. So some of the shapes and patterns in the turtle shell that I've been studying are the formation of these scales, the scutes that are covering the bony shell. You can see a little bit of these purple spots as they form and grow. They have interesting patterns that I've been studying in my research. So these scales are called scutes, as I mentioned. They're a type of skin appendage. They're these keratinized scales that form a tessellation over the turtle shell. On the left, you have the carapace. On the right, you have the plastron. And below, there are the early stages of development. You begin with this thickening of the epithelium overlaying the mesenchyme. And then in scute development, similar to scale development, you then have a flattening of this epithelium and then later keratinization. Skin has quite a variety in shape and form. So you are familiar with, quite likely, the scales that you see along the bodies of reptiles. And as I mentioned, these scutes on the turtle shell are a specialized type of scale. Feathers are a different type of skin, fur, and teeth. These are all different varieties of skin that are quite different in their adult differentiated forms. But if you go back to their developmental beginnings, there are a lot of similarities in the way these structures form. So you have these interactions between the epithelium and the mesenchyme, and you have signaling centers that are regulating their formation and patterning. I apologize that there's no pointer, but these purple spots are kind of are showing some of the signaling centers and how they're spaced in regular patterns in different systems. So one way I've been studying this is through the use of gene expression. So I've been studying the turtle embryos to follow the development of these structures. And gene expression is letting us know what types of molecular regulation is happening during the formation of these structures, but it's also a way to visualize them. So on the left, you have the beginning of the development of the turtle shell. And on the right, you have a slightly later stage when you're actually starting to form these scutes. And they have regular spacing between them. And we've been studying the dynamics of how this pattern and the spacing is coming to be. So the turtle shell is contoured by these signaling centers. And we've looked at several different molecules that we know to be active in the regulation of different types of skin structures, for example, hair, teeth, scales. And you can see that that these genes are expressed in and around these signaling centers. So BMP and sonic hedgehog have kind of overlapping expressions in these early precursors to these scutes, whereas gremlin seems to have, in this third column, has a complementary expression to those other genes. And using micro-CT, we also kind of studied how the signaling centers are contouring the shell. Looking at this in section, now we looked at kind of how the different expression patterns are separated and how they come together in the development of these scute precursors. Oh, I can't even see the mouse at all to move this, but at the top row you have BMP, sonic, and gremlin. And then looking at how these expression patterns complement each other, you can see that along the forming shell, there's a nested chain of these molecules, suggesting that they are controlling the spacing of these primordia, which then determines the patterning. So from observations about how these different primordia form, we started with some colleagues, Roland Zim and Isaac Salazar Ciudad, to try to explain this with a mathematical model. So let's see. Here on the right, you have the formation of the earliest of these precursors on the margins of the shell. So the thicker ones on the outside, and these are followed by the ones that are slightly more central. Then along the midline, you have another group forming. So there are three kind of different formations of different groups that are forming along the shell. And then in later phases of development, these spread and grow to have this confluence. So the dynamics of this formation can be described with a mathematical model that suggests that reaction diffusion dynamics are patterning these structures. So going from this pre-pattern, there are certain molecules that are expressed in the primordia, and there are molecules that are expressed around the primordia to then determine the spacing. And using these mathematical models, we can make hypotheses about how these structures are coming to be, which we can then go back to the lab and test ex vivo or in vitro. These very strange looking things are turtle embryos. So in the top, they're actually decapitated. So the heads are removed and the guts are removed. And then we're able to culture the embryos ex vivo and then do kinds of manipulations where we can try to get an idea about the molecules that are working to resolve this pattern. So on the left, we have control. I can't see at all where the mouse is, but I guess it's okay. There are regularly spaced primordia around this forming shell. So you have about 12 dots on each side. If we block a molecule called EDA, ectodisplacin, that is involved in development of skin structures, when you have defects in this signaling pathway, you quite often lose skin structures like hair or have malformation of teeth, fewer teeth. And when you block this pathway in turtle embryos, you lose the scute primordia. Similarly, another molecule, activin A, that's involved in TGF-beta signaling, so another signaling pathway. When you block this one, you get irregular patterning. So the primordia that were actually spaced out quite regularly now become more fused and irregular. And these effects differ in different embryos. So if they're slightly different ages, then the effects of adding this molecule on the pattern of the scutes are slightly different. But you still get this kind of irregular patterning, so that way you don't have the same periodicity to the precursors. We also looked at some other signaling pathways. So sonic hedgehog is a well-known one, and BMP signaling. These molecules that we had previously shown were expressed in the scute primordia. So in the center panel, if you block hedgehog or BMP signaling, you lose the scute primordia and you lose the contouring around the developing shell. On the right, if you block another signaling pathway, fibroblast growth factor, FGF signaling, and you get some effects similar to this activin A inhibition. And so you have this irregularity now to the primordia. So what this tells us is that these signaling molecules and their pathways are involved in orchestrating the signaling behind scute development. And using the model, we went back to see about the timing of scute induction. So there's an arrow pointing to sonic hedgehog, which is kind of the molecule that we're using to read the regularity of the patterns. So if the scute primordia are induced too early or too late, then you have different deficiencies. If they're inhibited too early, then you lose the primordia. If they're interfered with later in development, then you get more of this kind of striping and irregularity. We've also considered the development of scutes given the diversity that we have in turtle shells. So there's quite a regular pattern to scutes in turtle shells, but some turtles, like sea turtles, for example, have elaborated the scutes. They've added more, and some turtles, like the soft shell turtles, have actually reduced or completely lost the scutes. So this green at the bottom is the turtle that I've been using for most of the experiments. It's a red-eared slider turtle. It's a pest, invasive species that's quite easy to get lots of embryos for doing these kinds of developmental manipulations. But the Pelodiscus, circled in red, is a soft shell turtle. And in this group, Carapacial and Pelodiscus, they're keratoculiids. There have been instances of scute loss and reduction. So we looked at the embryos from these turtles as well. And so now this is a view where you can see the forelimb and hind limb on the side, and the heads are again missing, and those are to the right and the tails are to the left. So when we looked at the expression of these molecules that we know to have kind of regular patterning in the hard shell turtles, such as this red-eared slider below, in the soft shell turtles, we don't see any periodicity or any kind of these signaling centers in the development of the shell. And this is maybe not so surprising, but it's interesting to see that along this shell primordium, this carapacial ridge, instead you have a line of expression of these molecules. So the expression is there, but it doesn't actually get this kind of periodicity, this kind of patterning in the signaling centers. So a question there is, how did this come to be? Because presumably the scutes were lost in this lineage. So how was scute development more or less suppressed? And as I mentioned, there is little variation in the pattern of scales on the turtle shell. So these different groups on the outside and the left, there's this trichomy script of this red-eared slider. It has the most common pattern of scales on the turtle shell. So on the outside perimeter, there's usually 12. Down the midline, there are usually 5, and then on the sides, there are usually 4. As I mentioned, in some sea turtles, you have a few more scutes. But in all other turtles that have scutes, it's basically the same pattern as this red-eared slider. So loggerheads down in this Caretta species have an additional costal or pleural scute along the lateral margins. And then Ridley turtles on the right have even more scutes. So they have more down the midline and more on the, more in all of the domains of scutes. And so part of the point of this is that you have an increase in complexity in these sea turtles. But one thing that we noticed is that quite often you have abnormal variation in scutes. Even if most turtles have the same pattern as the red-eared slider, what we observe mostly outside of that, and outside of these sea turtles, is that there would be abnormal variation in the scute patterns. So these little stars and arrows are pointing out instances where you have extra scutes or you have a splitting of the different scute types. So we noticed this in our embryos, that sometimes the expression domains of these markers that we were using to study scute development were sometimes showing up in irregular patterns that were predicting what you might see when you have these abnormal scute patterns. And Roland Zim is the one who has been doing this mathematical modeling. He tried to replicate this using the model to see what could be the origins of this kind of abnormal patterning. And he was not able to replicate it with any kind of molecular genetic aberrations. And the only way that he was able to achieve abnormal variation was through the induction of a hemispheric offset, so that the kind of marginal scutes, which are the pre-pattern, were slightly offset. And then that should induce kind of aberrations in the central scutes, since those are forming later. And this is consistent with what we would think, given studies many decades ago of people just studying scute patterns from hatchlings, that this could be produced by something like mechanical stress, such as would be caused by desiccation, high temperature, environmental pollution, because many of these turtles have pliable eggshells. So in the picture on the right, you can see the embryo and its extraembryonic membranes. It's firmly attached to the eggshell. So any kind of mechanical disturbance that happens to the eggshell will actually impinge on the embryo itself. And we think that this is causing part of the abnormal variation that we see in the patterns on the carapace. These embryos are quite different than, for example, chicken embryos, where you have the embryo lying directly on top of the yolk. So we wanted to see if we could kind of test this hypothesis. And we worked with people who are actually working with these sea turtles. They were studying them for temperature-dependent sex determination, which turtles have. But at the same time, they are collecting a lot of data on the hatchlings. They take photos, they take measurements, and they too have noticed a lot of these abnormal patterns, and they were wondering what are the roots of the cause of these things. So on the left, you have the numbered pattern of like a normal number of scutes. And then in this middle and on the right, you can see some of the aberrations of the abnormal patterns that we've seen. And so this was together with Jeanette Weineken and Blair Bentley in Florida and Australia. And what we noticed was that under hotter and drier conditions, we see more anomalies in the patterns of scales than under normal and humid conditions, suggesting that this is possibly contributing to this abnormal pattern. But this is a complex story. It's not the only way that these very likely come up. I've heard stories from people who said that they've been working on some kind of tortoise farm and they had one tortoise with all the progeny having the same kind of anomalous scute patterns. So it's likely a complex structure that in some cases could have genetic and environmental contributions to it. So as I mentioned, I was going to briefly talk about links between the different projects that I've been working on. So I've been working on skin in general, and that is partly in the turtle shell and partly in teeth. So if this helps to understand the logic at all, on the left, we have this picture of this turtle carapace that's developing, and it has this regular pattern to the developing scutes. In the center, we have the same gene and how it's expressed in a developing tooth, in a molar tooth in a mammal. So you can see that the signaling centers are spaced apart from each other. And in both of these systems, a lot of the same molecules are actually determining the spacing between the signaling centers. In teeth, well, in turtles, again, you have the scales that are forming from these structures. In teeth, you have cusps, so the elevations that you have on your teeth. And this is shown a little bit in these purple spots. Those are the signaling centers in section. And then this 3D reconstruction is showing the cusps, so these bumps on the teeth that are going to be the final pattern from the development of teeth. And I started studying this because the lab experiments that I do are quite intensive. And sometimes you want to just go and look at something like easy, like morphology. I think that Alex can probably relate to this since he's also sometimes lab, sometimes paleontologist. And we have this incredible collection of fossils at the University of Helsinki that were collected by Alexander von Nordman, who's the first paleontologist in Finland, as before it was even Finland proper, but in Helsinki. And what we have in abundance are cave bear teeth. So cave bears were part of the Pleistocene megafauna that became extinct. Thinking about the kind of complexity that you have in the turtle shell and how it comes to be kind of inspired me to also start thinking about complexity in teeth. And I was just fortunate that we had this research collection because cave bears, as their name implies, spend a lot of time in caves, and a lot of the fossils that we have are from cave bears that died in caves. And the teeth are in incredible condition. So it's thought that these bears, there are a lot of hypotheses about how they became extinct. But one of them is that there was kind of some genetic instability in the population. So it was like a kind of decline in these bears. But their teeth are remarkable. They're incredibly complex, more so than most animals. And what I started doing in this collection was analyzing the morphology of these teeth. So this is a digital 3D model of the molar teeth, so the lower cheek teeth in the cave bear. On the left, you have the first molar, then you have the second molar in the middle, and the third molar at the end. and then you still have some remnants of the jawbone around that. And I have been thinking about the development of these structures, how complexity is generated along the tooth row, because the development of this structure has kind of consequences. The first molar starts, and then it's followed by the development of the second molar, and then followed by the development of the third molar. So I've been thinking about how this complexity is generated, and the starting point for that was actually just kind of imaging them and quantifying the complexity. So one component to this project was making these 3D scans and then looking at their complexity and comparing it to other bears. Bears are ecomorphologically diverse, so they have different kinds of species that are having different diets. Polar bears are carnivorous, brown bears are omnivorous, cave bears are presumably herbivorous, so presumably eating plant material because the teeth are so complex. That's generally the pattern that follows. If you're eating simpler foods, like if you have a carnivorous diet, then the teeth are usually simpler. And with teeth like this, they're so complex that it would lead you to think that they are most likely herbivorous. And there are other types of data that also support that, like isotope data. But the project that we've been working on is looking at this complexity and quantifying it and analyzing it from an evo-devo perspective. But this is actually going to take me back to the turtles. So to start analyzing these bear teeth, I was making these 3D models and quantifying the complexity. It inspired me to go back to the turtle shells and analyze them again. So turtle shells, even if, when they lose the scutes, the bony plates also have incredible complexity. And I started studying the bone sculpturing on turtle shells to understand kind of the complexity and also how they look differently when they have different ecologies. So you have different morphologies in the sculpturing, but then you also can see elements of scute formation or scute loss. And so this is something that kind of was the back and forth between the different systems. In one system, I started thinking about the patterning in terms of like the molecules that are forming the first shape, but then going back from tooth back to turtle, I've applied a different type of kind of ways to analyze it. And what we're doing with this is mostly considering the sculpturing in relation to the presence or absence of scutes, but also using this as a tool. So there are a lot of great resources for sharing these types of media, not just for studying them for research, but also as learning tools. It's very difficult to identify these different types of bones in the field if you're a paleontologist or if you're learning to become a paleontologist. So my colleague Pat Holroyd and I can't actually move this little thing to reveal her name, from the University of California, Berkeley, have been studying, scanning and studying these, so that way we can also kind of compile a learning set for people to use in education settings and in the field. But at the end, we'll also consider their complexity in a similar way to how I've been studying the teeth. So I hope the variety of these projects has been interesting and made some sense. And I thank you for your attention. And I wish I could find my mouse so I can figure out how to.
Thanks a lot, Jackie. That was very nice, very clear presentation. I'm going to open questions to our attendants. If anyone has a question they would like to ask Jackie. Let me see the.
Hi, I have a
Please.
question.
Please, Vicente, go ahead.
Hi, Jacqueline.
Hi.
Beautiful presentation. Thank you.
Thank you.
I'm not sure if you mentioned it, but the complexity of the patterns in the teeth and the scutes of the turtles reminds me of Turing patterns. I'm not sure if you have thought about that possibility.
Turing, yes. So that's, I think, the basis for some of these mathematical model with these reaction diffusion dynamics. Ultimately, it's coming back to these Gerhardt, Meyer Gerhardt. I don't remember the names exactly, but these people who applied, who studied Turing patterns and regular patterns that we see in animals, like zebra stripes and things like that. Yes.
Thank you.
Thank you.
Even fingers, right? There's this reaction diffusion thing, my friend.
Yeah.
And it's interesting though, the transition in bears, because they're going from something that I would perhaps consider, you let me know what you think, that's simplified, right? The ancestral pattern for them is carnivore dentition with the carnassial, which are basically blades, right? It's the premolar and the molars here, the last premolar and one of the molars, and which I guess have like 3 cusps. I don't know really. And then you have these bears that have, all of this complexity you've shown us. I don't even want to start counting cusps or anything like that. And it's really fascinating. The system would allow this. Usually what you think in evolution, simplification is like a one-way road. And this could be a very explicit example that that's not the case. And that perhaps just depending on the molecular signing systems that you're working with, it's very easy for them to go from simplicity to complexity, and it's not too complicated. And what do you think about that? Do these systems work this way, where you have like a couple of knobs and then you can just go from simplicity to complexity very easily?
Exactly. How does it actually happen? How is it kind of like modulated? Because you get more complex in bears, but then also the polar bear, the dentition, is simpler. I didn't show any of them here, but they're still way more complicated, more complex than you would expect for an animal that's actually eating meat, flesh. But still, they are reduced from, so the bears became more complex and then polar bears became less complex, whereas cave bears went the other direction, increasing complexity. So how these things came to be and how we could even understand how they came to be, I don't know. But I mean, at least we're kind of trying to understand some components of the puzzle, but how it can actually switch. And that's another thing, what I was talking about with these soft shell turtles. You had these scute patterns, and you completely get rid of them. Like how you kind of simplify so easily, or if you make things way more complex, how are these modulated? I don't even know if you're going to be able to get at this from a genomic perspective, or is it really going to have to be very likely by considering the development?
That's right. Yeah, I would say the developmental system is a key to understanding how these transitions are possible. And this makes me think, let's take teeth to scutes. Is there any way that, or is there any hint perhaps from phylogeny or whatever, that a soft-shell turtle may have recovered scutes in evolution? Or is this experimentally possible? Is there any hint of that whatsoever? Experimental natural variation?
I think not.
Okay.
I'm not the expert on that, but as far as I know, not. But we were interested in this Carapacial to the kind of greater group that has these soft shell turtles that completely lost them versus the ones that reduced them. Because Roland is the guy, like I mentioned, who's doing this mathematical modeling. And I was like, Roland, make the model, get rid of the scutes. And he tried and he tried and he couldn't. And of course, models are just, guesses, estimates. They're not going to be perfect compared to the biology. But the only thing he was able to recover was a pattern similar to these carapacialids, where the scutes are reduced.
Right, you do have turtles with reduced scutes, right? Like patches or something like that.
Actually, I think is it in the fossil carapacialids that you can actually see that kind of they look like they're fused almost. So you have fewer of them, and you have some losses, but a lot of reduction. But yeah, but it was just really funny that he couldn't actually recover this kind of scute loss in the model. So we couldn't actually understand the dynamics of how the molecules have changed to get rid of scutes. Well, you can do it artificially with the experiments, but I mean, that's not real. You can't just knock out these kind of pathways. And that's not how it would actually happen, because that would be really detrimental to the
It's informative about the system. It's informative about the system and the models too, because they say, well, you must be missing something. So you need to find that something. That would make it fit. Just one more comment and I will pass on Ignacio, who is a lifted hand. Because this losing the shell is happening in the Triassic with the first turtles, the Odontochelys, which still has teeth, and it even brought up a discussion about whether the scutes appeared later, or this is the non-shell, the primitive condition. But I would say it's the loss of the carapace. You have other stuff in the Triassic that do have it very early on. So the system must have been able to do this from very early point in evolution.
Yeah, this is what I was asking Tyler Lyson about, because I think people were thinking that the plastron showed up first, like the ventral part of the shell.
Odontochelys has a plastron, but no.
Yeah, right. But at the same time, when we think of turtle shell development, we always start with the carapacial ridge. Is it possible that it wasn't there, that you have this dramatic transformation without that major signaling structure? I don't know. I think we just can't tell from the fossils. It doesn't seem possible. And then this kind of decoupling of the different parts either. I think there are scutes on the plastron. I think he looked at that. Okay. But there's no evidence of anything from a carapace perspective.
Right.
Very strange.
Very curious. Okay, Ignacio, now you can make your question. Sorry to.
Hi, I'm doing some little identity theft because we were on the same lab. Thank you for the presentation, Jacqueline. It was very interesting. I wanted to ask you a little bit if you could give me a little bit more detail in how you are assessing structural complexity through your analysis, because I find it very, very interesting.
In some cases, I'm just using like very simple numbers. So like this plastron, this ventral part of the shell, has a few elements, and they're in like two rows, and that's it. Whereas the carapace has all of these different groups, and you have a greater number. So even in that sense, they're more complex, the carapacial scutes, just because you have greater numbers. And then you're more likely to see this abnormal variation in the carapace. And is it because of the way they form, or just by the sheer fact that you have more numbers, so you would have more kind of chances to have some kind of abnormal variation? So in that regard, it's just basic, like counting, the number of elements versus not. But then with the teeth, it's very challenging to count the actual number of elements. So the way that we're kind of analyzing it is by the number of faces or tools, like almost like a GIS, like if you would describe a mountain range, like how complex is the mountain range? So I'm analyzing the teeth from that perspective in terms of how many different faces are kind of being formed, which actually are forming the functional surface for what the animal is going to use for eating, for example.
Okay, and for the first approach, something that popped in my head, and because as I understand, there's, for example, for the number of elements, there's you were modeling by a reaction diffusion process, and I'm by no means expert in any of this, but my intuition tell me if you have a bigger space for a reaction diffusion process, like a bigger canvas, normally you will have more elements arising in the reaction diffusion process. Wouldn't be in that case that there's a increasing complexity that's only due to increasing size?
Yeah. One thing I didn't show was the sea turtles. Roland also tried to model how you actually get these additional elements. And it was just by stretching, basically, the area, by enlarging it.
So, yeah.
It just seems so simple, but maybe that's how it is.
Yeah, no, because I was thinking if that in that case, there is no shifting complexity of the underlying process, just a bigger canvas, so to say.
Yeah.
Okay.
I mean, you would think that the molecules have to have, I mean, there's a distance with which they're effective. So is there kind of like an increase in their expression to kind of maintain more territory or not? And just that you then are able to have kind of these like peaks and valleys of expression and then have more elements pop up. Maybe that's what happens in the end, and that gives more complexity.
Okay. Thank you. Thank you very much. It was a great presentation.
Thank you.
Okay, do we have anyone else that would like to make a question? Let me check the YouTube and see if anyone has dropped a question. Yes, here's a question. This is Marcia Gaete. Thank you for the presentation and check similarities between the pattern formation. Do you know how the morphological complexity is related to the final size of the structure? Thanks. Oh, kind of the Jose's question.
Yeah. This is actually, and at a conference, somebody actually asked this as well. But we had kind of used this as theoretical because we don't have access to sea turtle embryos. So in theory, we could actually maybe measure kind of the dynamics when you actually have something like that. Or if we had different species, the different species would actually be the same number of scutes. Anyway, we were thinking about this, but we actually haven't been able to actually to do that in practice because we've only, from the sea turtle perspective, for example, where you actually have more complexity and more elements, we just see them as hatchlings. We don't see them as embryos. Actually, but I was writing to somebody in South America when we were doing this project, trying to get, because they did have a collection of these loggerhead embryos, and I never got a hold of them. It was somebody who had a chapter, I think, in the Biology of Reptilia books, where they were describing the development of the loggerhead turtles. So then they maybe still had the embryos somewhere if somebody saved them to actually, because with the micro CT, where we could look at the contouring, we could scan those embryos and see the primordia. You can't really see them superficially. You'd have to section them or use some kind of this 3D imaging. And you could look at that and test the hypothesis, but we haven't been able to. We're not going to either, unless we find this, old collection from when people used to collect endangered species.
Hopefully you get lucky. Okay, everybody, thank you very much. Jackie, we're going to let you go to bed now, because it's pretty late over here in Finland.
Getting dark. I have to ride my bike home before it's too late.
Oh, my. Okay, good luck with that. Take care, Jackie. Thank you very much to you. And thanks to everybody to assisting. We will be seeing next Wednesday in another seminar.
Thank you for all of your questions. It was really fun.
Cool. Bye-bye.
Bye-bye.
Ciao, ciao.
Thanks.